Hello everyone, and thank you for attending this webinar on textural characterization. My name is Jeff Kenvin, and I will be the moderator for this webinar. Before we begin, we wanted to cover a few housekeeping items. At the bottom of your screen are multiple engagement tools you can use. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right corner. If you have any questions during the webcast, you can submit them through the Q&A. We will try to answer these during the webcast, but if we run out of time, it will be answered later via email. Additional materials are available in the resource list. We encourage you to download any resources or links that you may find useful. Some networks cause slides to advance more slowly than others, so logging off of your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. If you experience any problems during the session, you can find answers to some common technical issues located in the Help Engagement tool at the bottom of your screen. An on-demand version of the webcast will be available after today's webcast and will be emailed to you once the session has concluded. I am pleased to introduce today's speaker, Chris Gastaldo. Chris joined Micromeridics in 2012, and Chris is currently an application scientist and also leads the validation and testing of new instruments and software at Micromeridics. He has extensive experience with customer education, both in person and our online training programs. Chris has a wide range of expertise including physisorption, gas displacement pygnometry, particle sizing, and chemisorption. Chris, it is an honor for me to introduce you today. We are so glad you could be here to share your insights on sample grinding. What happens to surface structure when a material is ground?
Thanks, Jeff. I will be covering what happens to a sample when it is modified through a grinding in this webinar. The four materials used for this experiment are 13X, alumina, carbon black, and ibuprofen. The AccuPick 2 was used to collect skeletal density data, and the 3Flex and ASAP were used to collect surface area and porosity data. Each sample was analyzed 3 times to obtain averages and verify consistency. Before looking at the experimental data, we will discuss density, surface area, and porosity. Each of these topics are capable of their own presentation, so explanations will be kept brief. What is density? Density is defined as a sample's mass divided by its volume. The AccuPick 2 is a gas picnometer which is used to measure a sample's volume by gas displacement. Helium was used since it's the most ideal gas and does not interact with any of the samples in this experiment. This technique measures a sample's skeletal volume. The skeletal volume consists of the volume of the sample and closed pores, but excludes any pores that are open to the surface. The BET surface area model is the most common model used for determining a material's specific surface area. It assumes there are multiple layers of gas adsorbing, the surface is uniform, the adsorption energy of the first adsorbed layer is higher than all successive layers, and ignores all interactions between gas molecules. The most common porosity information are the total pore volume, median pore width, and pore size distributions. The pore size distribution models used for the analyses in this experiment are HK for micropores and BJH for mesopores. The 13X, alumina, and ibuprofen are solid materials consisting of no fine powders. The grinding process created particle sizes that do not exist in the original sampling. The carbon black is composed of a variety of particle sizes. The grinding process might have created particles in the lower size range or modified the structures of the existing particles. The 5 size ranges are the original non-modified sample, particles larger than 1.18 millimeters, particles between 500 micrometers and 1.18 millimeters, particles between 125 and 500 micrometers, particles less than 125 micrometers that were caught in the catch pan. The samples were ground, sieved, and separated prior to analyzing. This means the particles from one size range are not the same exact particles, ground finer in another size range. All materials came from the same lots. It is common to see larger particles in lower size ranges due to particle shape not being uniform. For example, an elongated particle can fit through the mesh depending on its orientation. Samples were pre-treated prior to analyzing, with the exception of carbon black and ibuprofen on the picnometer, since neither one of these are very porous or hold much moisture. Pre-treatment consists of applying heat for a specified time while under vacuum. The first sample we will look at is 13X. Here we can see the non-modified sample and all of the different particle size ranges. A scanning electron microscope was used to take images of the three lower particle size ranges. We can see the surface of the particles are not uniform and particles larger than the size range are present in the lower ranges. Density results are consistent across the different size ranges. Slight variations are most likely due to sample quantities used. The overall trend for analysis length does not appear to depend on the particle size. The analysis length increases as the sample quantity increases. Surface area and porosity results were impacted by the grinding process. 13X is composed of an alumina silicate crystal structure held together by a clay binder. The reduction of porosity within the mesoporous region is due to the breaking down of the binder's structure. This leaves behind an isotherm that resembles a type 1, which is expected of a zeolite crystal. The surface area results were consistent until the particles were broken down to less than 500 microns. Analysis length decreases as the particle size ranges become smaller. The analysis length for both the 125 to 500 micron and the sub 125 micron size ranges were reduced by over 70%. The linear isotherm shows the mesoporous region, and there is a clear decrease in porosity as the sample is ground. The surface area and pore volume results are impacted by this decrease. The logarithmic plot shows the micropore region. The trend shows adsorption quantity decreases as the particle size range decreases. One area of interest shows the two smaller ranges become saturated before the larger ranges. This is due to the nitrogen molecules having easier access to the micropores. A longer equilibration time or possibly changing the analysis gas for the larger particle size ranges could cause additional gas to adsorb in the lower range, causing the isotherms to shift and the overlays to be closer. The differential pore volume makes the shift in the distribution clear. As the particle size ranges become smaller, exposing the zeolite structure more, the pore size distribution shifts to a smaller size. The next sample is alumina. Again, we can see the non-modified sample along with the different particle size ranges. Here are the three lower particle size ranges for alumina. We can see the surface is not uniform and particles larger than the size range are present in the lower ranges for this material as well. Density results are consistent across the different size ranges, and the analysis length does not appear to be impacted by the sample quantity. Surface area is roughly 5% lower between unmodified sample and the lowest particle size range. Medium pore diameter slightly decreases as the particle size range decreases. The biggest impact grinding has is on the total pore volume, with almost a 9% decrease in volume. Analysis length decreases as the particle size ranges become smaller. The analysis length for slightly modifying the sample already shows a drastic reduction in time with somewhat minimal changes to results. The isotherms for the different size ranges are similar in shape and shift downwards, indicating a decrease in adsorption quantity. This makes sense since the pore sizes have slightly shifted and the pore volumes have decreased. There are negligible differences between the isotherms in the lower pressure region, indicating all changes happened in the mesopore region. We can see the pore volumes decreases with the decreasing size range. The particle size range of greater than 1.18 millimeters is sharper than the non-modified sample due to a lack of diffusion resistance into the pores. The next sample is carbon black. Carbon black is the only sample in this experiment in which the non-modified sample is composed of different sieve sizes. SEM images for carbon were not collected because the material is stuck to a carbon adhesive disc and it is not possible to distinguish between the disc and the sample. Density results are consistent across the different size ranges. The analysis length does not appear to be impacted by the sample quantity. Results for the larger size ranges trend downwards until the smallest size range. The unmodified materials results falls somewhere in between, which is expected since it's composed of particles from the different size ranges. The analysis length increased for the particles larger than 1.18 millimeter and sub 125 micron, and decreased slightly for the remaining range groups. The isotherms for the different size ranges are similar in shape and shift downwards from the largest to smallest size range, indicating a decrease in adsorption quantity. The isotherm for the unmodified material falls in between. There are no differences between the isotherms in the lower pressure region. The last sample is ibuprofen. The material within the coating is exposed when the sample is ground. Here are the SEM images for the ibuprofen. Density results are consistent across the different size ranges. Analysis length is trending upwards as sample quantity increases. The sample cup was filled to the same level for all the sample runs, but the finer material has more interstitial space between particles, resulting with less mass. Nitrogen and krypton surface area analyses were performed on the ibuprofen. Results are slightly different due to conditions in which the samples were analyzed and could have been improved by decreasing the free space volume for the nitrogen runs. However, it is not necessary since the interest lies in the trend that exists with both gases. No porosity information was obtained. The ibuprofen can be viewed as two different materials, the non-porous coating on the tablets and the porous material within the coating. When the sample is ground, the material within the coating is exposed, causing an increase in surface area. This explains why the results are higher than the ground sample. The surface area for the two smaller size ranges is less than the larger size ranges. This is caused by grounding of the non-coated material. What porosity exists in the material is being reduced, causing a lower surface area. In conclusion, what does grinding do? Density results were minimally impacted. The biggest change to these tests is the analysis length, which is dependent on the material. Grinding the material changes the amount of sample that can be placed in a sample cup due to the interstitial spacing between individual particles. How much of an impact grinding has with the surface area results depend on the material. If sample composition is not uniform, like the ibuprofen sample, the ratio of components exposed to the surface might change as the size ranges change. In the case of the carbon black sample, the particle sizes already existed in the bulk, so the impact was the sieving process and seeing how the individual ranges added to the results of the bulk. Porosity results vary greatly depending on the material. The biggest changes were observed in the mesoporous regions, including the total pore volume. Grinding materials allowed for easier access and diffusion into micropores. Analysis lengths were reduced as the particle size ranges decreased. This is the end of my webinar, and I'd like to thank you for your attention. We can open it up for questions now.